Thank you so much. I'm really happy to be here. I literally got off a plane from Abu Dhabi a couple of hours ago. So please forgive me if I speak faster than usual, which means it'll be breakneck speed because I was told I have 10 minutes. And so really you will be getting the Cliff Notes version of plant and animal genetics, but I'm very happy to share with you the full version, anyone that wants a copy of all the slides. Is this working? Oops. Oh, yeah. Obviously, I'm addressing all of Tom's challenges in one slide. So clearly, the reason that we focus on novel technologies, obviously, is one of the biggest challenges is global food security, as we have to feed 9 billion people by 2050, which means we need to literally double productivity, which in fact is rather an opportunity for Ireland. So I'll concentrate on some of those aspects. And given that this focus is on the technology, again, forgive me, this is a little technology heavy with the notion of how these particular technologies can be applied in Irish agriculture. And so my notion is pulling all the themes together and to say what we're looking at, really quite frankly, is smart plants, smart livestock, smart biomes, and smart environments. And sort of, I'm taking two approaches to this, and I think that's the way going forward, is sort of the holistic macro level, looking at all of the omic technologies in context, and then integrating into that a more reductive micro level, so that's modification and fine-tuning of the traits to basically have all of the components of the orchestra working together to get you a perfect concerto of crop and livestock production. And I know that sounds a lot rather grandiose, but really at a large level, that's what we're doing. We're trying to pull together the phenotypes, as in how our crops and animals appear, and what it is when you drill back down at the genetic level that allows you to select for the traits, the absolute traits that you want of disease resistant, improved nutrition, better characteristics for the animals and plants, and how you drill back to the genome. And by genome, I'm actually talking about the whole genome. I'm not just referring to the nuclear genome, I'm also talking about extrachromosomal organelle, microbiomes, all the components that make up the organism's whole set of information. And how we can marry those are really where we're focusing at the moment. And I guess how you would approach it is to look at from omics to improve crop and livestock. So we're trying to get that phenome gap down between the genotype, and again, by genotype, I'm referring to all of it, not just the chromosomal genotype that we all think about, to bring that closer to the phenotype, to the traits that we want in the crops and animals. And there is, of course, the whole omic cascade in between that we need to work on. And as Frank mentioned, this is all information technology. That's what life is information technology. So one of the most important tools we're developing is actually making sense of all of the biological data we're pulling out with the incredible technologies we're now beginning to develop to be able to analyze sequences. So we're trying to marry the genomics platform to the phenomics platform of improved livestock and crops through the macro level of looking at genomes as a whole, and then the micro level of going in and tweaking these specific components that will allow us to modify traits. So we're really looking at a synchronized molecular breeding, so a more precision type of breeding. But none of this will make any difference if we don't look at one major principle, meta-omic, and that's economics. If that doesn't make sense, the rest of it won't either. So it would be very nice if all the traits we were looking for would nicely map onto specific loci on our chromosome. So for ag traits, we can look at yield increase, stress tolerance, disease tolerance, both abiotic and biotic stresses, response to climate changes, improved nutrition, improved nutrient use by plants and animals, looking at better feed efficiency for animals, for example, better hardiness, reduced greenhouse gas emissions. But of course, nature is not that nice. most of those and many of those are quantitative trait loci. That means that there are several genes involved and they're turned on and off in response to various environmental stimuli. So we have to go about it in a rather indirect way. As was mentioned earlier, we're becoming really good now at sequencing cheaply. It also has given us a little bit of perspective when we realize the cow actually has more genes than we do. So cow is more complicated, and especially if you look at the microbiome, a cow is way the heck more complicated than we are. So that gives us a little bit of perspective. But of course, there's only so much you can do with the actual sequence, and it's really cool when you see right now the cost of a sequence is down to less than $1,000. There was an X Prize some years ago to be given to the first individuals that succeeded in getting, very thorough reads with, high density mapping of the, initially it was for the human genome, but they actually abandoned it some years ago because they saw they were getting there so quickly. But of course, as I mentioned, sequences are all very well and good. What you really look at here is the bottleneck, the phenomic gap, the correlation of the genotype to the trait that you're looking for. So it's the data analysis, the coevolution, if you like, between the statistical models, the sequencing, the high throughput sequencing and the high throughput phenotyping, and making sure we're not pulling out spurious associations. So the algorithms being developed by the silicon-based organisms here are as important as the actual data being produced by the carbon-based organisms. And if you look at the amount of terabytes, rather interestingly, Dublin is one of the places in Ireland in the world that's actually has the square footage to have industrial scale data centers. But of course, most of that is being used in financial analysis, not to help with Irish farmers. But here's something rather interesting that may help Irish farmers. They may all actually become their own citizen scientists. You now can get this minion, and it's not the little yellow minion. It's actually a handheld sequencer. You're possibly, within the next few years, going to have farmers out in the field doing their own sequencing. They might still they'll need a little bit of help on the analysis side, but they'll actually be able to do the sequencing. Of course, the real issue is turning that raw data into trying to make a determination of how these networks work. So this complicated strings of pathways, trying to pull that apart is a little bit more difficult than looking at the straight sequencing. And even if we start to bin those into interesting categories, through the various omics, from genomics, transcriptomics, proteomics, interactomics, metabolomics, all the way up to the phenomics, and we're developing much better tools on the IT side, we're still a long ways away. Frank very clearly mentioned the importance of one of the big projects that Ireland is leading the world in, and that's actually phenotyping cattle. As he mentioned, genomic selection has increased genetic gain by about 60% in dairy herds. Now, much of this was done in the US initially, as you can imagine. And of course, that's where the initial focus was. And if you look at the QTLs that are out there, most are associated with dairy traits. They developed, Teagasc did, a custom chip with Illumina. And this particular imputation tool, as he mentioned, will so that Ireland will soon have the largest set of genotyped animals in the world, up to, I believe, heading towards 1 million now. Of course, the big thing, again, there's 250 trillion data points that are going to have to be analysed. What this allows you, of course, is with marker-assisted breeding, you can determine breeding value at birth. So now you can much more quickly increase, selection intensity, reduce the generation time, and increase the rate of genetic gain. So you're looking at potentially developing speciality herds for specific types of traits. And as he also mentioned, for the animal's own sake, you actually are heading into the area that's of very important, great importance to humans, and that's personalized medicine. You will also have personalized animal health. So each animal will have its own barcode determining exactly what you needed to feed it to get optimum output. That's not really going to happen, but and this is just looking at some of the QTLs that are showing up here for sheep. You see, not a lot, but they're the obvious ones you're looking at. For chicken, rather more, and number 1 up there is egg weight. Again, not surprising. Next is abdominal fat, etc. You look for pigs. I was a little shocked to see the highest QTLs for pigs was drip loss. Anyone know what drip loss is? I'm going to the Teagasc people here. That's actually the amount of, in processing, the amount of moisture that's actually lost from the meat. And needless to say, the one that has the largest and expectedly is cattle. And I think from the trend here, you can see exactly what I mentioned earlier. The initial focus very much was on dairy herds. So the look was for milk fat and all of the characteristics that you would be looking for when selecting for specific high value, high merit value animals for breeding programs in dairy cattle. Now, here's the way to seriously speed up this whole process. And actually what you can do, instead of going ahead and having physical animals, you can produce multiple animals at the same time from banked frozen fibroblast cell lines. Do you think this might work in Ireland? But it's a really good way of looking for high generic merit, because you're not going to be spending the amount of money you would do on purchasing and managing recipient females, so that you can maximize value by selecting and transferring embryos. So now you're way back before the calf with the desired genotypes that you're looking for. So this for sure is the future. Perhaps a little bit of a hard sell yet in Ireland, but perhaps the day will come. Now, if you're interested in more than just the particular traits that you can find within your herd, and you want to actually go in there and do this for plants and animals, we have a massive, big toolbox of traits that are available. Trait modifiers, shall we say, that are available at this point in time. And I will pick out the ones that I think have a potential in the short period to work in Ireland, and you probably can guess the reason that I will pick those ones. But I'm not going to go through them all, but as you can see, many of them deal with the whole RNA family, and in fact, the power tools that we have in there, the genome editing, it is actually again based on RNA, using this as a mechanism to go ahead and target the specific areas that you're trying to modify. And I'm genuinely referring to these genome editing nucleases as power tools, because they're incredible. I'll give you examples in a moment. But there's also other really interesting ones, such as like trait up. This is for plastid transient expression operons, where you basically turn on whole suites of genes, and you can have it turned off again by the time it goes for commercialization. So you can't see whether it's been modified or not because it was transient. You can use tunable, they're developing now totally synthetic tunable promoters and transcription factors. So for example, a promoter that will just respond to a pathogen when it's attacking either the animal or plant, and so you don't have a genetic load of having it been expressed at all points in time. There's a relatively new target that's been found, target of rapamycin. These kinases are master regulators in everything from yeast up through plants all the way up to humans, and they turn on whole suites of transcription factors. So it's a master modulator of a master modulator. There's this relatively new switch system that's just been worked on now, called PPR. These are dynamic switches for endogenous genes. So you can track or relocate endogenous RNAs. You can alter sequences and coding capacity. You can put tags on them so they go to specific sites and you only get the expression in that particular site. There's a bunch of others, and you're going to hear specifically from Fergus as he talks about epigenetic modifications, especially at the the microbiome within animals and also within plants. And you heard earlier regarding synthetic biology, you can literally now go to BioBricks, and it's actually called the BioBricks Assembly Kit, and build your own complete circuit of genes that you want to produce whatever in your particular organism. This one down here, I think we'll be waiting a while before you'll get EU approval for any of that in your farm animals or plants. But speaking of, there is a way for Ireland to be interested in next generation trait modification, because you can circumvent the whole GMO issue. Now, it kills me to have to say this, because as a scientist, I actually consider much of this as much to do with language editing and using terms that are not statistic. Well, they really wouldn't stand up at all in a scientific symposium. But you play the game. Oh, this is being taped. If you want to get through the regulatory process, and these are sort of 4 or 5 specific groups that are potentially will not be considered, and they're not being considered in the US by US regulatory agencies as transgenic. Jury is still out a bit in Europe, but not totally, interestingly enough, especially when we look at these, as I called them earlier, the power tools of genome editors. These are what the EU are looking at, some of the very specific examples that they're looking at. And rather interestingly, last year, they came out to this company, Teagasc, who produced this canola seed that's resistant to sulfonylurea. This is a herbicide tolerance. And they used an old, far more clunkier method than CRISPR to actually produce this sulfonylurea tolerance. They went to Germany, and Germany told Teagasc, yes, we will consider this as conventional plant breeding, even though it has been modified with this editing tool. Now, as I said, the work that these guys are doing, for example, they're looking at potato for phytophthora resistance, of huge interest to Ireland, and bear in mind, Germany said this is not genetic engineering, but quite frankly, the technology has supplanted what they were working on, and pretty much everything that they're doing now, they've abandoned their system and they're moving on to CRISPR. So this are the set of synthetic nucleases, and the ones that are going through the process because it takes so long to go through deregulation process. Most of the ones that are out there right now are zinc fingers and talons. But as we've been hearing all morning, the one that's going to be the future is CRISPR. And this is what Bruce Whitelaw said. Some of you know Bruce. He's at the Roslin Institute. He said, genome editing basically provides the variation you want where you want it. And to be honest with you, I'm referring to CRISPR as the Swiss knife of genetic modification. It even looks a little bit like this. You can do bespoke genome engineering. You can modify, customize this particular nuclease to do anything almost. That's a little hyperbolic, but believe me, it's amazing. Every day you hear something new that's been done. You can use it, as I just mentioned, to knock out genes, so to knock out sensitivity genes or anti-nutrients, whatever your particular interest is within the specific organism. You can enhance specificity by these nicases, where you're just making single strand nicks in the strands. You can do very precise modifications. You can do insertions, obviously you can do deletions, and I'll give you an example of a few in a moment. You can use activation or repression of target descriptors, so it may potentially eventually replace RNAi. You can use genome-wide screens using these CRISPR systems. You can use it as a sensor, so for example, you could do very rapid TB testing and get your result bang right there with a very sensitive response. Now there's one area that is of somewhat concern, and this is the notion of using gene drives to drive whole modifications very quickly through species. So clearly, guess where they're looking at doing that right now? And this is for the Zika virus in mosquito, the Egypti mosquito. There is a concern there. I am very concerned about anything that says mutagenic chain reaction. And that's what they're calling it. So I'll tell you some examples of where they're looking at making sure that this isn't going to be a problem. But if you look at the literature just within the last 2 years, it is enormous. It is taking off exponentially. Patents are being applied every day. Between when it was first put in for the initial patent by Jennifer Doudna in 2010, it is literally running off the chart, and the amount of money that's flying into this technology is just unbelievable. Just give you a few examples on the plant and animal side. And here, the very first examples were done in tobacco, sorghum, rice, and Arabidopsis in 2013. That's the first time they succeeded in using CRISPR to use modifications in there. And since then, a ton of work has been done. I'm just going to pick a few here. Here is a real value of using this to modify, for example, wheat, because of course wheat is a hexaploid, but this is some work that was done by Dr. Wang to make wheat resistant against powdery mildew, which has been a real problem because you were dealing with the hexaploid in the past, but here he was going to be able to alter all of the homoalleles, bang, right at once. Here, this work that was done in John Innes. Now, this was important because they were looking at something that is a real concern for sure in genome editing, and that's off-target edits. And you might have heard some very scary story recently about a mouse that had inhaled a virus, and the gene editing went to work on the virus that was inhaled to give it syncytial virus respiratory problems. So this is something you have to be careful about, and you really, really need to worry a little bit to make sure that this tool is well managed. And speaking of, if you go to the Iowa site, you can design your own target sites, and they will make the CRISPR for you. And here's just just a few examples from the animal side. Again, remember, it takes time to get through the regulatory process, so these are still talons, they're prior to CRISPR. This is some work that was done by Recombinetics here. They introduced just 212 BPs from the Angus sequence into a Holstein bull and got hornless Holsteins. This is very important in dairy herds in the US. Roslin, they took a few amino acids from the warthog, put it into the RIALA gene in swine and made them resistant to African swine fever. Here we have chickens that were modified to turn off the ovalbumin gene, again to help with allergenicity. And this is a comment that was made by Stock Farnberger, who's the CEO of Recombinetics. He said, an animal could be edited to have the very best genes its species can offer. The genome is information, and this is information technology. We have been able to read the genome for a long time, now we can write to it. The other thing Scott has said, he's going to bypass actually talking to the FDA, and he's going to say, well, Germany said Cibus's canola was not genetically modified, so our animals are not genetically modified, and I'm just going to release them without regulatory authority. I hope that was hyperbole. And here are some other examples, and yet another person, another individual who's very prominent in this area, Kevin Wells, he said, these pigs, the receptor for this PPR respiratory virus was modified to basically turn it off with the CRISPR. And he said the pigs challenged in this study do not contain any transgenes. And he is correct. The CRISPR was used to switch off the susceptibility receptor and made them resistant to this respiratory virus. George Church, who was one of the founders of this technology, used this to turn off to inactivate all 62 porcine endogenous retroviruses in the pig genome. Of course, his interest is for xenotransplantation. It's not for agriculture use. The notion is to have pigs as potential sources for xenotransplants for human organs, primarily heart. Chickens have been modified to basically to go two different directions. As I was talking about speciality herds, here you can specialize chickens to be female only for the egg producers and male only for the meat. So you can have a CRISPR chicken. Sorry. And here we have what I'm calling, I've coined this myself, the Schwarzenegger phenotype, where they've modified the myostatin gene, which basically controls the production of muscling. And now you have Schwarzenegger pigs, Schwarzenegger cattle, and Schwarzenegger lambs. Sorry, that isn't PHR, but it is using CRISPR to turn all of these off. In Davis, my colleague Valison van Eeneman is editing the SRY gene, which determines maleness. And what she's doing is basically turning these XX females into male appearance. So the genotype is still XX female, but the phenotype is male. She's also controlling sterility here to make sure that this is considered a cisgenetic effect and it's not going to escape. My concern, as I mentioned earlier, is one of the most powerful capabilities of CRISPR is gene drive, to basically drive complete mutations very, very quickly through a population. And of course, the concern with that is you can seek of all the positives. You can drive these systems to destroy or modify animal and plant pests, noxious weeds, invasive species. But you do not want this to escape for sure. And this is where I personally would say, let's have a little caution before we look at this CRISPR disruptor in a larger context. I'm going to obviously let Fergus talk specifically more about the microbiome, but just to mention, plants also have microbiomes. And in fact, the component under the soil, where you have mycorrhiza and various bacterial, in addition to fungal sources that are a huge part of the whole microbiome of a plant. And in Davis, we've actually discovered a particular landrace of maize that has co-evolved with these diazotropic microbes that basically allows it to fix nitrogen. This is massive. Here you have a monocot fixing its own nitrogen. And Fergus is going to talk about the phenome, I presume, looking at modification, epigenetic modification through, for example, nutrient intake in the animals. So this is the diverse types of phenotypes you could guess as reproductive performance, disease susceptibility and resistance, increased productivity, growth rate, etc., in response to the food that is modifying, say, for example, methylation sites, as in this example of the Gaucher mice here, exactly the same genotypes, quite different phenotypes based on feeding regimens of the mother. So be careful what your mother eats while pregnant with you. That's a bit of reverse engineering. So going forward, I'd see the challenges are the technology itself, obviously, quite frankly, the application, especially the regulatory environment, acceptance, and we often think acceptance by the consumer, but the first acceptance has to be by the farmer. If the farmers hadn't been so involved in the whole genotyping experiment that Teagasc have just conducted, it just wouldn't happen. So your first point of sale has to be the farmer. And then, of course, the practicality and economics of the implementation itself. So some of the low-hanging fruits, as I mentioned earlier, is the system biology modeling of the systems and processes. So looking at improved genome-wide analysis, better computational tools that will allow us to have a better understanding of these complex regulatory and metabolic pathways. Then a systems approach for discovery and decoding pathways, and then using these new tools to tweak them. Better genotyping and feeding, so we have better predictive biologies of indicator traits, and better indicators that actually lends themselves to automation, so we can get harmonization of data from these automated systems, so we can have standardization, better ontologies, high repeatability, very precise trait modification to integress into these elite systems then, and then the tuning expression with tunable promoters and tunable transcription factors. The higher fruit, and the one that's way the heck off, I would think, are the synthetic biological systems. So now you can go in to BioBrick, buy your bits and pieces, build this assembly kit, produce artificial chromosomes, operons with large multigenic constructs, synthetic inducible promoters, so that, as I said earlier, there's one that's been done in plants, in microbes with this phytosensing that can detect explosives in the environment. Then you can use this to construct these artificial biological systems and devices that exhibit predictive behaviors. Putting together parts to get what you want. So this is interesting from a scientific perspective. It's a way off before any of this can be approved, no matter how perfect it may seem. But most important, to focus on orchestrating networks, whether it's the subcellular network of genes interacting, the networks of populations of organisms, and the networks of organizations, whether it's farmers here, but also international organizations, because there's a ton of data from all over the world. And yes, of course, some of those countries are your competitors, but there's also a huge opportunity for actually working with these individuals to compile all of that massive data we have out there, and to develop systems for pulling out the valuable information so we can apply it to futuristic crop and animal agriculture. So thank you. Probably 2 minutes.